Okay. In this video, we are going to discuss about the cytokinins. So in previous videos, I discussed about auxins, gibberellins. So in here, this is also a kind of a plant growth regulator. So in here, this one I mentioned in the previous videos as well, because when we consider about this cytokinins, gibberellins, and the auxins, so unlike in the case of abscisic acid and the ethylene, we are using S prefix at the end of the word, cytokinins, gibberellins, auxins. So the reason behind this is actually, so when it is come to these three plant growth regulators, these are not one compound, these are group of compounds. So therefore, we have to make the word plural. So therefore, that is why we are adding S prefix at the end of this word. So with that knowledge, let's move on to the next slide. So in here, if you consider about the meaning of these cytokinins, here the cyto, you know that cyto means cell, and the kinins it's refers to as the division. So therefore, the whole meaning of the cytokinin is the cell division. So when it is come to the the sites of the sites of synthesis, so it is mainly synthesized in the root tips. So and additionally, it is synthesized in seeds, fruits, especially the developing fruits, the fruits that are fruits that are being developed, and the and actually not developed, actually the fruits that are developing, the developing fruits, we can see a lot of cytokinin inside the fruits and also the leaves. So but mainly the cytokinin can be seen inside the root tips. It is synthesized in the root tips. So this is kind of a survival mechanism. So what do you mean by the survival mechanism? So usually the cytokinin is responsible for formation of the shoot system of a plant. So let's if you can let's say when a plant is in a natural condition, so the let's say the plant the plant is exposed to, the plants are exposed to different damages, many damages. For example, if it could be a human damage, or else it could be a animal grazing. So therefore, because of all these things, the sometimes the the shoot system can be removed from the plant. So if the cytokinin is present in the shoot system, so now the shoot system is no longer available in the plant. So therefore, um, no longer available a place to synthesize the cytokine in the plant. So therefore, the shoot system will not be grown. So as an adaptation, so the plant have therefore luckily the cytokinin is produced in the root tips. So therefore now the root tips are available. So therefore the root system is available, the root tips are available. So therefore the cytokinin is synthesized in the root tips. So therefore this cytokinin stimulate the formation of the shoot system. So this additionally the cytokinin is produced by seeds, fruits, and the leaves. Okay, now let's shift our attention for early experiments in relation to the cytokinin. So in 1955, the identification of the kinetin was done by the Miller. So actually, this kinetin is not present in the plant. Actually, those days, there were many experiments in relation to the tissue culture. You know, those days, those are the days that the tissue culture was coming to the picture. So what we do in tissue culture, actually we take a piece of leaf, and after that we try to we are multiplying plants by using that particular plant tissue or the plant part. So by using an aseptic condition. So in here, so scientists did many experiments in relation to the cell division. That means, however, they wanted to multiply the multiply the cells more quickly. So therefore, they added many things, many artificial compounds, and as well as natural compounds to growth media. So one instance is herring sperm. So the herrings are this kind of a fish. The sperms of these herrings was used as a compound. Actually, the herrings sperms, they incorporate the herring sperms to the growth media in order to see whether the cell division is increased or not. So therefore, usually any compound, before we incorporate into a growth media, so we do autoclaving. So because any substance, before we add into the growth media, so we have to sterilize it. So in order to sterilize, we have to place them in the autoclave. So likewise, the herring sperms also was placed inside the autoclave, and after that they incorporate the herring sperms into the growth media. So after that, they saw higher rate of cell division. So, and later they named it as kinetin. Actually, this kinetin is present neither in plants nor the herring sperms. Here, actually, what has happened in here, the adenine molecules which are in the herring sperms has been converted into the kinetin as a result of exposing to high temperature and high pressure during the autoclaving. So after that, in 1963, the Letham, he's the one who identified the natural occurring cytokinin, which is this creatine. So actually, the creatine, it was extracted from sea maize. That is why he named it as creatine. Sea maize, the maize or corn. So in order to be a natural occurring cytokinin, I mentioned this one in previous videos as well. It should be widely spread, or it should be distributed throughout the plant kingdom. So therefore, after that, he identified that this creatine is present in many of the organisms in the plant kingdom. So therefore, this is a kind of a natural occurring cytokinin. Okay. Next, we will discuss about the physiological effect of the cytokinin. So the first thing is the delay in the senescence of the leaves. So what do you mean by this delay of the senescence of leaves? So here, what do you mean by the senescence of leaves? Sometimes you might have seen like the plant leaves, they become, usually most of the plant leaves are green in color, and in some leaves of the plant, we can see in yellow in color. So, and after after that yellowing, the shedding of the leaf is taken place. So what do you mean by this shedding? So we call them as abscission. So in here, usually, let's say when a leaf is immature, so the leaf are incapable of producing own food. So therefore, they cannot do the photosynthesis. So therefore, they totally depend on the mother leaf. So let's say when it is matured, so let's say when a leaf is matured 30%, so it starts its photosynthesis. So when it is, then after that means it will become 100% mature leaf, and after that the senescence will be happen and abscission will be happen. That means shedding of the leaf will be happen. Okay, so this senescence process actually this senescence process is very common in temperate countries. So senescence process is happening in all plants, but in a particular season, the senescence processes happen in temperate countries, right? So you might have seen this in actually autumn season. In the autumn season is after the summer and before the winter. So all the leaves in some temperate trees are fallen down. For example, if you look at oak tree in the autumn season, so you will see that leaves become yellow in color, red in color, and orange in color, and after that the shedding of the leaf is taken place. Leaves are taken place. So this is the senescence, not the shedding, the yellowing, and that part is called as the senescence. It's kind of an aging process. So here basically what happened, if the when the leaves are removing from the plant, it is a waste stage for the plant because leaves contain nutrients. The removing leaves means the removing nutrients from the plants. So therefore the plants have adaptations for overcome this problem. The one such adaptation is that, so usually when a leaf is going to senesce, so you know that inside the cell we can see the vacuoles. These vacuole membranes are broken down. So you know, inside the vacuole we can see a lot of enzymes. So the enzymes which are responsible for breaking down of carbohydrate, proteins, and nucleic acids, and so on and so on. So here, so since now the vacuole membrane has already been broken down, so now the enzymes are come out. So it will be come to the cytosol. So in the cytosol, we will see many carbohydrate, complex carbohydrate, proteins, as well as nucleic acid, and also many other things. So therefore, these complex compounds are converted into its simple forms. For instance, if you consider about carbohydrate, the carbohydrate will be converted into sugars because of that enzymes, carbohydrate digestion enzymes. So the other thing is proteins will be converted into the amino acids. And also, if you consider about nucleic acids also converted into its digested products. So likewise, so the complex compounds will be converted into simple compounds. Yes. So, but and also I forgot to tell you that the chloroplast membranes are also broken down, and inside the chloroplast we will see the chlorophylls. So the chlorophyll also will be broken down. So therefore, what happened? The chlorophyll is the pigment that is responsible for the green color of the leaves. So that is why the green color is disappear and the yellow color is appear, because now the chlorophyll is broken down. So now the carotenes are there, carotenoid pigments. So the carotenoid pigments are going to appear. So therefore, the plant will become yellow in color. So, but this mitochondria does not break down until the end, because you know that even though it is senescence, the leaf is senescence, so still it is living, so still it is attached to the plant, so it need energy. So in order to produce the energy, so the leaf need the mitochondria. So therefore the mitochondria is not broken down until the end. So at last the mitochondria will be broken down, then the shedding of the abscission of the leaves will be happen. So this process we call them as senescence of the leaves. So why I said all these things, so when it is come to this senescence process, so the cytokinin is responsible for the delaying of this senescence of leaves. Okay, here, let's say if you detach a leaf from a plant, so after some times, after few days, it will be senescence. So, but if the leaf is not detached from the the tree, so the senescence process will not be happen in that speed. That means the senescence process will be delayed. That means when the leaf is attached to the plant, the senescence process will be delayed, and when we detach the leaf from the tree, the senescence process will be happen in a fast manner. So therefore, the reason for this is actually when the plant is attached, when the leaf is attached to the plant, the cytokinins are available in the plant. So therefore, it's delay the senescence process. And when we detach the plant, when we detach the leaf from the plant, so now the amount of cytokinin present in the leaf is low. So therefore, here it is the senescence process will happen in a fast manner. If I give you another instance, if you consider about, let's say, we can propagate the African violet or the begonia or the peperonia plants by using leaves, right? By using leaves, we can propagate peperonia, African violet, and the begonia leaves. And so, but if you detach a leaf from an African violet or a begonia plant, so if you keep it as it is, so it will be senescence. But if you plant that particular leaf in the growth media, for let's say a sand media, so after few days, that means after few days, if you look at after few days, the plant the leaves will be remain as it is. So because now the root, before it give rise to new plantlets, the rooting is happen. So what happened when the roots are formed? The roots are the major sites of cytokinin synthesizing the cytokinin. So therefore, now the leaves contain these roots. So therefore, the root produce cytokinin. Because of this cytokinin, the shoot system will be promoted, will be developed. So that is why it is not senescence. Actually, it's delay the senescence. So what this cytokinin do actually, when you consider about aging process or the senescence process, in here the cell membranes are broken down. So mainly cell membranes are broken down by the free radicals. So in these free radicals, the formation of the presenting of this cytokinin prevents the formation of this free radicals. So that is why, therefore, it prevents the aging. No, it's not the prevent. It's delay the senescence process. So here, basically, so this can be applied for our scenario as well, because, but not cytokinin. That is why people say to eat more food that contain antioxidants. That is why people say to eat. People recommend to eat more and more fresh foods in order to delay the aging process. What happen in our aging process? The skin gets wrinkles. So why the wrinkles are happened? Wrinkles are occurred because of the breaking of the damaging of the cell membranes. Because our skin also has been made out from this. Skin also contain cells. So the damaging of the cell membrane is taken place. Therefore, cell integrity will be damaged. So therefore, that is why the aging process will be happen. Wrinkles are formed. So therefore, if you these are also happen because of the free radicals. These free radicals are destroyed by the antioxidants. The formation will not be happen when there are more antioxidants. So that is why it is recommended to eat more the fruits that contain antioxidants. That is why people usually commonly people say that to eat fruits to become to have a beautiful skin. So that is the reason. So the other thing is the stimulate the cell division. The stimulate cell division. So it is, you know, that the stimulating the cell division, you know, when the Miller incorporate what do you call the herring sperms to our the growth media, he identified that huge amount of cell division. That means high rate of cell division. So therefore, the cell this is in terms of kinetin, but this is true for all cytokinins. So the other thing is to stimulate the development of lateral buds. You know, when it is come to the auxins, the auxin causes the apical dominance because it avoids the growing of the lateral buds. But this cytokinin, it stimulates the developing of these lateral buds. So therefore, the branching will be happen. So the other thing is to stimulate the germination of some seeds. So you know, when we discuss about gibberellins, we discuss that the gibberellins causes the gibberellins cause the breaking down of seed dormancy. Therefore, subsequently, it will be the seed will germinate. So here also, they stimulate the germination of some seeds. So the other thing is causes the nutrient flow around the plant. So what do you mean by this nutrient flow around the plant? You know, the places where the cytokinins presence have the ability of the absorbing or the taking the nutrients into that particular place. So that is why the fruits, the growing fruits contain higher amount of cytokinin because growing fruits need nutrients, especially carbohydrates. So therefore, now the cytokinin is present in the growing fruits. So therefore, the growing fruit now has the ability of taking up the nutrients from the other part of the plant. So therefore, the growing fruit take nutrients, including carbohydrate, from the other part of the plant. So therefore, the fruit will be developed further. So you can do a small experiment. Actually, it's not a small one, but if you select 2 leaves from one plant, so and you treat one leaves with the radioactive carbohydrate and the other leaves with the cytokinin. So if you keep it for a while, not a while, let's say a few days. So after a few days, you will see the most of the many radioactive carbohydrates are present in the cytokinin treated leaves, not the radioactive carbohydrate treated plant leaves. So therefore, so it's indicate that the leaves that present cytokinin has the ability of absorbing or the taking up the carbohydrate. So you can do another experiment. This is very simple. So in here you have to detach a leaf from a plant and apply small drop of cytokinin to the plant, plant leaf. So here the place where we apply cytokinin remains as it is in the green color. So, but the rest of the parts will be converted into the yellow in color, because the place where we had cytokinin, it's delay the senescence. The other places are senescence. So that is the reason. Okay. With that, let's move on to the uses of the cytokinin. In here, we are using cytokinin in tissue culture. So I discussed this in the auxin video as well, because when you consider about cytokinins, when you consider about tissue culture, so in the tissue culture, it is important. So what we do in tissue culture, we take a small piece of small part of a plant, let's say a piece of leaf, and we convert into a callus, and from the callus we take a shoot system and a root system. So first what we obtain here is that a shoot system, the shoot. After that we obtain the roots. So you know, at the very beginning of this video, I mentioned like the cytokinin is responsible for the shoot development, and the auxin is responsible for the root development. So since at first we are trying to stimulate the shoot development, so we have to incorporate more cytokinin than the auxin. So the auxin to cytokinin ratio now low. At first, the auxin to cytokinin ratio is low because cytokinin amount is high. So therefore, auxin to cytokinin ratio is low now. After that, when we need to develop a root system, so we have to incorporate more auxins to the growth medium. So therefore, now auxin to cytokinin ratio is high. So therefore, therefore this cytokinin used in tissue culture in such a manner. Okay. The next thing is to delay the senescence of cut leaves, cut flowers, cut foliage, and the vegetables. So here, you know, in good old days, the carnations are the #1 cut flower in Sri Lanka. And let's say there are many cut flowers, roses, carnations, chrysanthemums, and here you can see alstroemeria. So this is alstroemeria. Even in Sri Lanka, we can see the alstroemeria, but in cold places like Nuwara Eliya, Bandarawela, we will see. We can see this alstroemeria. It's available in red color, in 3 colors. As I know, red color, pink color, and the white color. So here, so by using the cytokinin, we can increase the shelf life of these cut flowers. That means we can delay the senescence process of these cut flowers. So therefore, it is very important in exporting and also selling locally. And not only this cut flowers, but also cut foliage. For example, in croton. So in order to keep the leaves very longer period of time than naturally, so therefore we can apply cytokinin. So not only this cut flowers and foliage, but also it used for the vegetables as well. So these are the main uses of the cytokinin in commercial uses. So therefore, this is the end of the video, and we will meet from another video soon.